Yeah, I was invited to come to the ISH to talk about inherited or germline predispositions to leukemia and other blood cancers. And so I gave a presentation about that, talking about some of the new knowledge that have come from genetic studies that have been done in those unfortunate people who develop leukemia or other blood cancers in the context of a familial inherited predisposition. We don't really know how many patients are affected because we don't really haven't looked deeply at the number of patients who have mutations in there that they inherit that lead to them having an increased risk of different blood cancers. I would say that we know most of the mutations that cause what we would say is a high penetrance cancer syndrome, that is where a brother and sister, or two sisters might be affected, or a brother and a first cousin. But we don't know if people who are developing leukemia or other blood cancers in their 40s and 50s have some genetic alterations that they are born with that make them more likely to develop those diseases at a fairly young age. We've known some of the genes in some of the more common syndromes where we could recognize that a patient had a particular disease that was associated with a risk of leukemia. But what's really changed in the last little while, a few years, has been in families where there hasn't been an obvious explanation, where everybody appears pretty normal when they're born and don't seem to have Down syndrome or Fanconi anemia or some other known abnormality, but nevertheless there's a strong family history of leukemia. By just sequencing the genomes of those patients, we've learned some of the mutations, and perhaps not unexpectedly, a lot of the genes that are involved tend to control very important processes in blood stems, hematopoietic stem cells, and bone marrow cells. What's really changed in the last few years has been using these high-throughput genomic sequencing technologies to identify the causative gene mutations in some families where we really didn't know what they were before. And this is primarily in families where there's a normal appearance in the individuals and no known genetic disorder, but they clearly have a familial leukemia or cancer predisposition. And the genes that have been turned up most commonly has been GATA2. Another gene that's been turned up more frequently has been RUNX1, and finally CEBP alpha, all known genes that are very important in controlling the normal developmental switches in hematopoietic stem cells as they develop. And so maybe not surprising that these sort of master regulatory genes of gene transcriptions might be involved. It's interesting that these tend to be loss of function, and we don't really understand why losing one copy or one allele, a mutation in one allele of GATA2 or RUNX1 may account for this. The other thing that we've learned is particularly in the GATA2 families, we've learned that a number of different familial disorders or hematologic cancer and immunologic disorders that we had previously thought were distinct are actually caused all by GATA2 mutations. It's clearly a moving field and a moving target. I would say that one of the things that I think that clinicians need to be aware of now is that if a patient presents with myelodysplastic syndrome, say before the age of 40, there's now pretty strong, compelling evidence that that patient likely has some sort of germline predisposition. And that can be important for that family, for their brothers and their sisters. It can be very important for patients and their families who are still in the reproductive years. They may be thinking about having additional children, and obviously if they know they have a leukemia mutation. I think also if there's a strong history of a first-degree relative, a sibling or a first cousin with leukemia, these should also be considered for genetic screening. Finally, another very important aspect of clinical practice is we obviously can't eradicate the mutation in an individual who has a RUNX1 or a GATA2 mutation. But the treatment of choice for these patients is bone marrow transplantation, frequently, particularly for the patients with MDS or AML. And obviously the donor we would think about most commonly using would be a brother or a sister or a cousin. And in a familial cancer syndrome, you don't want to transplant the bone marrow from a sibling who has the same genetic mutation into a patient who has MDS or AML, as those cells, first of all, tend not to engraft very well, and second of all, they're obviously also predisposed to the disease. So it's important for clinicians who are considering transplantation in these patients to be aware that younger patients with MDS or AML there should be very careful family history and perhaps genetic testing of them and of any bone marrow transplant donor. So I'm a pediatric hematologist oncologist, and we actually, about almost 3 decades ago now, identified one of the more common predispositions to leukemia associated with a familial disorder, and that's neurofibromatosis. And we were surprised at the time to discover that the neurofibromatosis gene controls the output of another protein called RAS, which is a very important cancer gene. It is mutated in about 1/3 of all human cancers. And so we've worked for the past 20 or 25 years to understand how mutations in the RAS gene, the NF1 gene, and other genes in that pathway contribute to leukemia, and how we can use drugs to treat them, targeted drugs. I think the most important take-home message for clinicians and researchers would be, first of all, I think one of the major research questions is, now we know what these mutations are, but we really don't understand why, for example, losing one copy of RUNX1 or AML1 makes patients predisposed to leukemia and myelodysplastic syndrome. And I think that's a very important question, because these genes are frequently mutated somatically, that is, by not germline mutations, in patients with MDS and AML. And I think the other take-home message is that clinicians should have a high index of suspicion for a familial component to the development of MDS or AML in younger patients, and that genetic counseling and genetic testing is becoming, and diagnostic tests are becoming increasingly more available for families and can help inform clinical management, particularly around bone marrow transplant.